The events that lead to muscle contraction occur in three general phases: the events at the neuromuscular junction, excitation-contraction coupling, and cross-bridge cycling. In this learning objective, we will look at each phase in detail. So when we think about muscle contraction, it all happens really quickly and generally appears to happen without too much thought or effort. However, when we start to break it down, there are actually many moving parts and pieces to muscle contraction that it's amazing that it happens so seamlessly most of the time. Now, when I think about all of the events that need to take place for muscle contraction to occur, I like to think of it happening in three general phases. Phase 1 is the events that occur at the neuromuscular junction. And the neuromuscular junction is just the junction or the synapse between a motor neuron and a muscle. Here, an action potential travels along the motor neuron and then moves across the synapse to innervate the muscle. The second phase is excitation-contraction coupling. And this occurs when the action potential has crossed the synapse and reached the sarcolemma or the plasma membrane of the muscle fibre and causes the release of calcium within the muscle. And then phase 3 of muscle contraction is cross-bridge cycling, and this happens when the calcium that has been released during excitation-contraction coupling moves tropomyosin away from the myosin binding site on actin, and the myosin and actin can actually bind, forming cross bridges, which allows the muscle to contract or to shorten. So beginning with that first phase, the events that occur at a neuromuscular junction, and in this image we have a motor neuron, we have a synapse or a synaptic cleft, and we have a muscle fibre or a cell, which can also be called a post-synaptic cell because it's post or it's after the synapse. All of these structures combined form the neuromuscular junction. So the first event that will happen at the neuromuscular junction is that the action potential will travel down along the axon of the motor neuron to the axon terminal. This change in membrane potential will cause our voltage-gated calcium channels to open. This allows calcium to enter the neuron, moving down its concentration gradient. Calcium in the axon terminal signals to our vesicles full of neurotransmitter. This causes the vesicles to move toward the plasma membrane facing the synaptic cleft. These vesicles will then merge with that plasma membrane and expel that neurotransmitter via exocytosis. The neurotransmitter diffuses across that synaptic cleft and binds to our ligand-gated sodium channels on the sarcolemma or the plasma membrane of the muscle fibre. So putting that into a written summary for you, an action potential travels along the motor neuron to the axon terminal. The change in membrane potential causes our voltage-gated calcium channels to open. Calcium enters the axon terminal. Calcium signals to our neurotransmitter-filled vesicles to move toward the membrane. Vesicles merge with the plasma membrane and release the neurotransmitter via exocytosis. The neurotransmitter diffuses across the synaptic cleft, and it will bind to the ligand-gated sodium channels on the muscle. Remembering, ligand-gated channels open in response to the binding of a chemical. The second phase of muscle contraction is excitation-contraction coupling. And the term excitation-contraction coupling describes the rapid communication between the electrical events occurring in the plasma membrane and the release of calcium from the sarcoplasmic reticulum, which is what actually leads to the muscle contraction. So in this image, and it looks a little bit different, we have our axon terminal, which is carrying our action potential. We have our synaptic cleft, and then we have our muscle fibre, which is surrounded by our sarcolemma. So our first step in excitation contraction coupling, which actually involves a few of the events at the neuromuscular junction, is our action potential travels down that motor neuron and triggers the release of the neurotransmitter. At a neuromuscular junction, so at the synapse between a motor neuron and a muscle, this neurotransmitter is always acetylcholine. The acetylcholine will then bind to our ligand-gated sodium channels on the sarcolemma, or the plasma membrane of the muscle cell. This allows sodium to enter into the muscle, and the action potential runs down through the muscle. So our action potential spreads along the sarcolemma and down into our T tubules. This causes the release of calcium from our sarcoplasmic reticulum and the terminal cisterns in our sarcoplasm. Calcium then binds to troponin, which is, remember, this blue structure on our thin filaments. That causes the tropomyosin to move out of the way and expose the binding sites for myosin. So here is our thin filament. A reminder that the yellow dots are our actin, this brown line is our tropomyosin, and the blue is our troponin. So calcium binds to that troponin. The troponin changes shapes and moves this tropomyosin out of the way. So now the myosin binding sites on actin are now exposed. Because those myosin binding sites are now exposed, our myosin heads can now bind to actin. They then pull the thin filaments toward the center of the sarcomere. And it's this that is considered muscle contraction. So the thin filaments moving towards the center of the sarcomere causes the muscle fiber to shorten because the two ends of that sarcomere are getting pulled closer in towards one another. The action potential will then eventually stop, and calcium is actively transported out of the sarcoplasm. Because there is no longer calcium bound to our troponin, it will move the tropomyosin back into place, and those binding sites on actin will no longer be exposed. So our cross bridges will detach and causes our muscle to relax. So once more, a text summary for you, and don't stress if this doesn't make any sense just yet. This phase in particular is one that we will go over again and again in the labs. You might also like to go over this a few times on your own. But we have an action potential arrives at the axon terminal and causes the release of acetylcholine. Acetylcholine binds to the ligand-gated sodium channels on the muscle, and then sodium can enter that muscle. The action potential then runs along the sarcolemma, which remember is the plasma membrane of the muscle, and down into the T tubules, which causes the release of calcium from our sarcoplasmic reticulum and the terminal cisterns. Calcium binds to troponin, which moves the tropomyosin out of the way and exposes the binding sites on actin. Myosin binds to actin and pulls the thin filaments towards the thick filaments in the center of the sarcomere. This is the part that shortens the muscle fiber or the muscle cell and therefore causes muscle contraction. The action potential will then stop and calcium is transported out of the sarcoplasm. When this happens, the troponin moves the tropomyosin back into place, blocking those binding sites on actin. Now, the final phase of muscle contraction is cross-bridge cycling, which does actually occur during that excitation contraction coupling phase, but it's explored in more detail when we just talk about cross-bridge cycling. So this figure is describing the process of cross-bridge cycling, but instead of focusing on the release of calcium and troponin moving tropomyosin out of the way, we just look at the formation of the cross bridges themselves. So a cross bridge is just when the actin and the myosin heads bind. So firstly, the myosin heads hydrolyze ATP and become reorientated and energized. What this means is that the myosin heads contain an enzyme which breaks down ATP, and the breakdown of ATP causes the myosin heads to move from this position back into this position, which we now call reoriented or energized. From this position, the myosin heads can now bind to actin. So this is our myosin head. Remember, the yellow is our actin. When the myosin and the actin bind, we call this forming a cross bridge. The myosin heads then rotate towards the center of the sarcomere, so from this position to this position here. It pulls that thin filament towards the center of the sarcomere, and it's sometimes termed the power stroke. This is what shortens the muscle fiber and is what we actually call contraction. Now, the power stroke is fueled by the release of ADP, or adenosine diphosphate, which is formed when we break down our ATP. To cause the release of cross bridges and therefore the relaxation of a muscle, a new ATP molecule actually has to bind to our myosin head. And now this part does seem to trick people. We actually need ATP to bind to myosin for the muscle to relax and not for the muscle to contract. And this is why when people die and ATP is no longer produced, the body will become stiff and go into a state of rigor mortis, because no new ATP is being produced to attach to the myosin head and therefore cause the detachment of the cross bridge and the relaxation of the muscle. And then, of course, this whole process is called cross bridge cycling. So this whole process will continue to occur for as long as the muscle cell is being depolarized, calcium is being released into the sarcoplasm, and we have ATP present. So a written summary of our cross bridge cycling. An enzyme on our myosin heads will break down ATP, and that head becomes reorientated. Myosin heads bind to actin, forming cross bridges. The myosin heads then rotate towards the center of the sarcomere, which is what we call the power stroke. It's what shortens the sarcomere and causes muscle contraction. Then ATP will bind, a new ATP molecule will bind to the myosin head, causing myosin to detach from actin, for that whole process to either happen again or for the muscle to relax. Now, just putting all of that together in one big hit, and I will go over this quickly, but please come back to this in your own time. So at the neuromuscular junction, we have an action potential moving down the motor neuron. This causes a change in membrane potential, which opens our voltage-gated calcium channels. Calcium then moves in to that axon terminal, signals to our synaptic vesicles full of the neurotransmitter acetylcholine. It signals these vesicles to move toward the plasma membrane, merge with that plasma membrane, and then expel that neurotransmitter or the acetylcholine out via exocytosis. That neurotransmitter or acetylcholine will diffuse across the synaptic cleft, bind to our ligand-gated sodium channels, which can also just be called the receptors, and this allows sodium into the muscle cell. The last phase of our neuromuscular junction, moving into our excitation-contraction coupling, was that action potential running down the motor neuron, causing the release of acetylcholine and the binding of the acetylcholine to our ligand-gated sodium channels. That causes sodium to rush into the muscle and the action potential to run along the sarcolemma and down our T-tubules. That then causes the release of calcium from our sarcoplasmic reticulum and our terminal cisterns. The calcium binds to troponin, so binds to these blue bits here. That pulls the tropomyosin out of the way, now exposing those myosin binding sites on actin, which are our yellow dots here. Now those binding sites are exposed, our myosin heads and our actin can bind. They form a cross bridge, and this thick filament pulls the two thin filaments closer towards the center of the sarcomere. This causes muscle contraction. Eventually that action potential will stop. This muscle cell will stop being depolarized, which causes the calcium to be actively transported out of the sarcoplasm. The troponin will move that tropomyosin back into the way of that binding site. Myosin and actin will detach, and the muscle will relax. Looking at our last step, which was just that cross-bridge cycling. So we have a breakdown of ATP on the myosin head that causes the myosin head to move back into this position, which is reorientated and energized. The myosin head can now bind to the actin, forming the cross bridge. The myosin head then pulls forward, so it moves into this position, pulling that thin filament closer to the center of the sarcomere. This is where we have muscle contraction. We then need a new ATP molecule to bind to the myosin head for our myosin heads and our actin to release.